Hello, everybody. I hope you are doing well wherever you are listening to this, and I appreciate your time. So I will be talking today about some recent updates to NCBI Virus, including a tool called the SARS-CoV-2 Variants Overview. I'm going to spend a few minutes on why it's worthwhile to spend our efforts on specialized resources for viruses. Then I'll present some of the improvements we have made to facilitate access to SARS-CoV-2 sequence data. So after over 2 years of living in an international pandemic, you can't have escaped hearing about viruses. But what exactly are they? They are extremely small, for one. You cannot see most of them unless you have a specialized microscope, and they require machinery inside of a host cell in order to replicate themselves. Viruses have a core of nucleic acids, which is their genome, and that's surrounded by a protective capsid. So they kind of look like a popular candy that doesn't melt in your hands. The genome can take many different forms, which is why it can complicate how we store and analyze the genomic sequence data. Understanding viruses is crucial to human health. It's estimated that 3 out of 4 emerging human diseases are from animals called zoonoses, and most of these are viruses. This is not a problem that is going away anytime soon. In fact, scientists predict an increasing risk of zoonoses through increasing contact between wild animals and humans or their livestock, which creates opportunities for spillover of pathogens, so spillover from a natural host into a human host. Humans travel, and that can lead to faster spread of new pathogens to wider geographic areas. Also, climate change can expand the range of insects that carry diseases. Several coronaviruses have historically been zoonoses, and coronaviruses have a wide variety of animals as their natural reservoirs. So this is a phylogenetic tree showing the relationships between various groups of coronaviruses. Some are responsible for mild but common diseases like a cold that you might get in the winter, and others cause much more severe infections like Middle East Respiratory Syndrome or MERS, and of course, severe acute respiratory syndrome, SARS and SARS coronavirus 2. Importantly, it seems that several of these viruses, including those related to SARS, exist in the wild as a group of closely related but diverse viruses, meaning that there may be more viruses that are not recognized but have the potential to cause severe human disease. There are many ways to try to understand viruses, how they spread, and predict which ones may cause disease or why some of them cause more severe disease. Studying their genomic sequence data is one of those ways. Genetic data carries the instructions for building the tools that give viruses and other organisms their abilities and their characteristics. NCBI Virus is a suite of value-added resources that provide sequence data, and they currently feature SARS coronavirus 2, but all viruses are available through NCBI Virus. Most people don't necessarily want every single publicly available sequence record. So we have a variety of filters that narrow down your data set based on normalized metadata. We also are big fans of using visualizations to explore data. And downloaded data can be in the form of genomic sequence or individual open reading frames. And we have flexible download menus to minimize the amount of extra work that you have to do to clean up the data before you continue working it. So what kind of data can you get from NCBI Virus resources? Well, we pull in the sequence data and the metadata that describes it from GenBank. That includes data from INSDC partners based in Europe and Japan. We also pull in some data from Bioproject and Biosample, which are descriptive records for sequences. And then we're starting to pull in data from SRA, which holds raw data or sequence reads that are usually assembled into the genomes that you might find through GenBank. And we're also doing taxonomy for viruses, and we create RefSeqs that are reference sequences for a particular species, the sequences that you can go to see what the genome tends to look like or find important proteins that might be on that genome. We worked with Eric Nowacki, who's a researcher, he's in the research branch of NCBI, and also the GenBank submissions team to develop a tool to annotate and validate sequences as part of the submission process. The viral annotation diviner, or VADR, can be used to annotate the positions of genes and the proteins on a variety of sequence types. This makes data more useful because you can identify the tools that a virus codes for. It helps to ensure data quality, and it significantly reduces the processing time for submission of a virus and release to the public. Submission of a virus sequence and release to the public. All of these proteins are also, they're stored in NCBI Virus, so you can either access information for the entire genome or for the individual proteins. And for various reasons, data descriptors in NCBI databases can be quite variable. So as a data consumer, normalized metadata makes it possible to build data sets that are relevant to you. So we have a semi-automated curation process. Once a sequence record is publicly available, we pick it up usually within an hour or two, and we normalize various fields. So for example, the isolate source field describes the part of a host where a sample was pulled from, and it can contain terms like serum, PBMCs, plasma samples, and so on, that we match up to one single term, which is blood. We also create and maintain the viral RefSeqs. So as I mentioned, there's usually one RefSeq per species, so they're useful as examples of high-level viral diversity. But when there is an important virus that is in the human population, we also tend to make a RefSeq for that too. So we have a RefSeq for the original SARS virus from, I think it was around 2003. And we also have a RefSeq for the SARS-CoV-2 virus that emerged recently. One of the benefits to having a specialized resource for viral data is that we can respond quickly when there's an epidemic or a pandemic. So this is a screenshot of our NCBI virus search interface. It incorporates a variety of filters that can be applied to the sequence data and a table showing your results and the normalized metadata. We developed several new features to support accessing SARS-CoV-2 data. So every day we check for updates to the pangolin tools, which is a common tool for classifying the lineages of SARS-CoV-2. And we run that pangolin classification on all SARS-CoV-2 sequences every night. The classifications are available through the NCBI Virus Results table, and if you download the FASTA or the metadata table, including the pango field, it will automatically include the versions of the pangolin tool, so you know which version of the tool was used to get those results. You can search for viruses from a country or from a US state, newly released sequences, or randomly sampled sequences, which should be more representative of lineage frequencies out in the wild. You can work with either genomic sequences, proteins or CDS, or amino acid sequences. And while the main NCBI virus results table only provides GenBank data, so only the assembled sequences, you can still search using SRA accessions, bioproject, or biosample accessions, which are from the same sample as the GenBank record. So it helps to work across the different NCBI databases. Once you've narrowed down your data set, you can explore the results through a multiple sequence alignment, which highlights the mutations and displays some of the metadata. You can also quickly generate a phylogenetic tree from your sequence data. However, both of these tools can only be run with a limited data set of about 500 sequences, but it's a nice way to be able to verify that the data set that you're working with is what you think it is. We also provide flexible download formats. Sequence data can be downloaded as FASTA files, either the nucleotide sequence, just a coding region, or a protein sequence. And the metadata included in the FASTA def line is customizable. You can also get a list of accessions, or you can download a table of the normalized metadata describing your data set. Okay, I'm gonna switch topics here just a little bit. Over the course of the pandemic, you've probably heard a lot about vaccines, antibodies, and spike proteins. But here's a short overview. Spike proteins decorate the outside of a virus. When they come into contact with a receptor on a host cell or on a human cell, they act like a key, opening a door and allowing the virus inside the cell, and then it can replicate and lead to disease in that host. So through natural infection or through a vaccine, your body can develop antibodies that stick to the spike, kind of like putting tape over a key so it can't fit in the door lock anymore. But the antibodies only stick if they can recognize the spike protein. So the spike has to look like the same protein that the antibody was created for. So if the virus develops mutations that change the way that the spike looks to the antibody, then the antibody may not work as well or at all. So in response to the SARS-CoV-2 pandemic, the National Institutes of Health initiated a collaboration called ACTIV, which stands for the Accelerating COVID-19 Therapeutic Interventions and Vaccines, that's ACTIV, Tracking Resistance and Coronavirus Evolution, TRACE, initiative. Our primary goal as part of this collaboration was to support accurate sequence analyses needed to track the mutations and prioritize emerging variants that may not bind well to antibodies or which may not be as treatable through the therapeutics that we have available. So once identified computationally, those variants can be studied in wet bench experiments, and we can promote sharing of the genome sequences with researchers and with public health groups. And because we were prepared for large-scale analyses, the team decided to go with a cloud-hosted pipeline, then to deliver the results of that pipeline to a web page hosted on premises. We also decided to use raw read data in addition to the assembled sequence data that's in GenBank. The raw sequence data is held in the Sequence Read Archive. You may have heard that called SRA. Because it's not averaged to generate a genome sequence, the data allows identification of major and minor variants. These records are also extremely large, so it's convenient to take advantage of technical tools like cloud storage or machine learning to do analyses. And then we can share the results with a wide variety of people who may not have access to those technical tools. Besides the amount of data, another challenge that we had was how to identify the groups of viruses that may warrant further investigation. So people refer to SARS-CoV-2 lineages like Pango that you're probably familiar with. Those are names like B.1.617. And they're grouped at fairly high resolution. So sequences within a Pango lineage are pretty similar to each other. This is also the lineage, the classification scheme that's most used in research and epidemiological studies. For our purposes, though, it had a huge detriment, which is that the pangolin classification tools can't process unassembled sequences. So we would lose all of that unique data sets that are in SRA. So a team at NCBI developed a method for assigning Pango equivalent lineages through sets of defining mutations. So we have curators who identify the mutations that are required for each lineage, and then each mutation must be present in a particular sequence in order for it to get classified as that lineage. We've also done some studies, so we verified that the way that we are assigning the Pango classification has very, very high correlation with the way that sequences, like whole genome sequences, are assigned using the classic pangolin tools. The pipeline we ended up with was pretty complicated. It involves many people and several different teams at NCBI, as well as outside collaborators. So in one step of the pipeline, sequence data from GenBank and SRA go through a validation step. Contigs are built for unassembled data, and then mutations are identified by comparing to the RefSeq. In a separate second step, the mutations identified in sequences are compared to the lineage-defining mutation sets, and then sequence records get classified as Pango lineages. Finally, the data is aggregated with sample metadata, such as the collection date or location, so it can be served up to accessible cloud directories or our variants overview website that I'll be talking about more in a minute. So we have all this data that's coming out of the pipeline, literally millions of data points. A researcher interested in the data could get lost among the many files of output, and like the saying goes, they might get lost in the trees and not be able to see the shape of the forest. So if you've been to the NIH campus, or perhaps you've seen a map like with the trees and not the roads and everything highlighted, you might recognize this area though, but most people, even if they're familiar with an area, probably won't recognize that top box just because it's overwhelming, even though it's still accurate. If you label the items that are here, you start to become more familiar with what you're actually seeing. So it becomes more understandable, but you still maintain accuracy. But if you're only looking for one particular place, it's still too much information to be useful. So you might want some kind of highlight on only what you are interested in. Our goal with the SARS-CoV-2 variants overview was like that. We wanted to summarize the potentially overwhelming sequence data and help customers find the data that they're most interested in, all without losing any accuracy. The SARS-CoV-2 overview, the SARS-CoV-2 variants overview is an example of how visualizations help convey the stories in the data. Over the past couple of years, it's emphasized that people involved in the pandemic response are often severely short on time. Groups may not have access to a bioinformatician. So we could help to support public health efforts by providing information on mutation-based lineages and their spread. So this helps to support pandemic response, public health needs, getting information out to people as quickly as possible. And the information that we're using for this is really developed through global efforts to sample and sequence SARS coronavirus 2. So people who are going out there and doing the swabs and collecting the samples and really have put tremendous effort into making this data publicly available. We identified common questions that a researcher or someone in public health might have. We address these questions through our SARS-CoV-2 variants overview webpage. This is an interactive dashboard that summarizes the where, when, and who of pango lineage selected by the United States CDC. So this page only includes variants of interest, variants being monitored, variants of concern, and variants of high consequence if we're ever unfortunate enough to identify one. As mentioned previously, this resource is unique in that it seamlessly combines unassembled and assembled sequence data from SRA and GenBank, as well as the sample descriptors from BioSample and BioProject. We also use the BioSample data to identify when a single sample is represented by data in GenBank and SRA, so that sample isn't counted twice in the data that we're showing to customers. And if you're interested in trying this out, you can go to the URL that's shown there, or you can search NCBI Virus in most web browsers. When that page comes up, you can click that button the button highlighted by yellow, the variants overview button. Okay, so our first user story that we identified was what mutations are associated with the lineage and what risk factors might be associated with that lineage. So all of these lineages with a CDC status are listed on the left of the web page, highlighted there in yellow. Customers can search by pango lineage, WHO Greek letter, or by CDC status. And each lineage has its own little info panel showing the lineage-defining mutation set that was developed by the curators here at NCBI. The info panels can also be expanded to show key mutations, or in this case, variations, because we use the amino acid positions for the mutations. These are in epitopes or binding sites identified by our protein domains team. We also link to web pages developed by another active trace participant called NCATS, and they've gathered data on the effectiveness of therapeutics against various SARS-CoV-2 lineages. An info panel can be clicked to select a lineage, and then the other displays on the page will highlight how often the lineage is sampled and in what locations. Several members of Active Trace are from pharmaceutical companies, and it's important for them to know which lineages are growing in the countries which use their therapeutics so that they can be sure that their therapeutic will still continue to work against the lineage. So we show the frequency of the CDC status lineages over the past several months and use a metric called doubling time, or how quickly we expect to have twice the current number of records to highlight quickly growing lineages. Pop-ups allow customers to dig into the data and see actual counts as well as frequency of records, which helps with interpretation. Another question many people are interested in is which lineages have been detected in my state or my country. When no lineages are selected, the US states and countries are colored to show the number of samples classified as a lineage with CDC status. And if a lineage is selected, then the maps only show the counts for that lineage. Clicking on a location also generates a pop-up with lineage counts, the total number of samples, and the number of lineage records collected over all time. And although the current version of the SARS-CoV-2 variants overview addresses several customer needs, we do have plans for many different improvements that we'd like to make in the future. So the teams began collecting and reviewing usage data from logging on the page, presentations to audiences, and user interviews. We also would love to get your feedback. So there's a yellow feedback button that stays in the bottom right of the web page when you access it. You can just type in your comments there. So we're making data-driven improvements to the resource. And we know that customers are interested in being able to see all the mutations associated with the lineage and mutation on individual mutations in general. So we'll be adding that in the future. We also hope to enhance the options for downloading the data, since many people would like to be able to perform their own analyses on this valuable and unique data set. And to finish off, I just wanted to say I am sincerely grateful to the many people who have contributed to the work that I presented today. There's several teams at NCBI highlighted on the slide, and I'm personally privileged to work with all these talented and hardworking people. We also appreciate the work of our external collaborators, the people collecting samples and submitters, because none of this would be possible without their efforts. And finally, thank you for your time and attention. And I am very happy to take some questions if there are any.
Thanks, Anita. I'll give people a chance here to put some questions in the pot. I don't have any open ones right now, but I have a couple of questions. So in terms of the sampling of the and what we're getting into the database, how has that changed over time? And are we still getting, adequate surveillance of what's out there in the wild right now? Because it seems like with the current, if you're thinking about the United States, with the current resurgence, it seems to me that everybody that I know is getting COVID, but those people are not getting their DNA checked. They're just basically doing an at-home COVID test, positive, they isolate for a few days, and mostly they're okay. But, it seems to me like we're not getting as much of that data in as we once were. Is that the case? Or how does that work?
Yeah, the amount of the sequencing rate, the submission rate has dropped. So we are receiving fewer SARS-CoV-2 sequences. However, there are some federally funded national efforts to kind of randomly sample from the population. So the amount of sampling that happens really depends on what kind of questions need to be answered at that particular time. So early on, we just needed to see, where is the virus at this point, right? Remember when it was, is it in Washington State or is it in North Carolina? And then it kind of changed to, how is the virus evolving? What kind of mutations is it picking up? What does it look like compared to the antibodies developed against the vaccine? And so that's a lot of the sequencing that we've been seeing over the past several months. Recently, though, sequencing efforts have kind of tamped down, because being able to do this general, lower level, but across the population sequencing lets us identify any potential viruses that have mutations that could be more risky, that might allow for escape from antibodies or from therapeutics. I'm always a big fan, I think the more data that we have, it's always better, but obviously there's funding that goes into it. It has to be justified to answer the questions that people are asking now.
Thanks. And I have a sort of a random question that has always bothered me, and that's this this pangolin name. What is the origin of that? Because remember early on, pangolins were considered to be a possible source for SARS-CoV-2. Is it just a coincidence that it's called pangolin?
No, scientists are funny sometimes. We try to make jokes. So early on, because it was one of the things, we thought that it may have originated in pangolins, and I'm not sure that that's been completely excluded or not, but it's an acronym that could be used for the classification system. So I can't remember what it stands for, but it is an acronym for a longer classification name.
But it's more than a coincidence.
Just something.
It's more than a coincidence.
Yeah, it's more than a coincidence. Yes.
That they called it pangolin. Okay, that's
maybe tongue in cheek.
Right, I understand. But so they sort of came up with an acronym that fit that animal's name.
Yeah.
Because of the initial interest in that. Okay. And so Rana has put in the chat actually what that acronym is. It's Phylogenetic Assignment of Named Global Outbreak Lineages. And so you can make that into pangolin if you leave that.
Yeah, and I do have to say, most of the classification systems that we have for other viruses, it works a little bit different, because we have sparse collection, right? So if there's an outbreak, you might only get a handful of sequences.
Right.
But with this outbreak, we have a really dense set. So you have very, very few mutations when comparing from one sequence to another one. So it really was important to have a classification system that could handle the density of sequences that we were getting.
Yeah, and I mean, in some ways, this is an unprecedented natural experiment where we have so many copies of this virus out in the world. And
yes, absolutely.
This opportunity for evolution. So we do have another question, an actual question from a participant. This person asks, are there any plans for an API or other programmatic access to NCBI virus? Or maybe does that already exist?
Yes, okay, so there's a tool that currently exists called datasets. So you can just search for NCBI datasets. That's only for SARS-CoV-2 data or coronavirus genomes, but they have very good explanations of how to use the datasets tool either through command line or through API to access the data. If you have any trouble with it or if for some reason it doesn't satisfy your needs, please reach out to NCBI because we would love to hear like how we can make improvements to our tools.
And I've put that the URL, direct URL to datasets in the chat, but that's a good point, and that's a way to get access to lots of different kinds of genome-related data. And there's a special package for SARS-CoV-2 that you can get from there, right?
That's right. Yeah, I know in the future they have plans for adding access to all the virus data that we have in NCBI Virus. So they are using like the normalized metadata that we're showing on our resource too. That's in the future though. For now, I think it's limited to mostly SARS-CoV-2, and there are some things that you can pull for coronaviruses in general.
Yes, and if you go to the datasets homepage and you just scroll down, there's a whole section on SARS-CoV-2 genomes, SARS-CoV-2 proteins, and there's some explanations of how to do things with those resources right there on the page. Okay, so thanks everybody for coming, and thank you very much, Inida. And one thing that came up a couple of times in the chat is everyone would like to have your slides. So if it's okay with you, I'll go ahead and if you send those to me, I'll put them up on that FTP site for people to get access to. No,
okay, yeah, perfect. And thanks for the opportunity.
Sure, yeah. Great to have you. Bye-bye, thanks.